Yay. Cool. Okay, so we had our timers go off. And this is like pretty hot, but it's touchable. Let me see. Just want to show you what I was talking about. Yeah. Yeah. So that's how you know you have cleared the roots. Yes, absolutely. So in the methods, all of these methods you could do without heat by just letting them sit overnight in the solutions. The only reason we didn't do that because we wanted you to see every step, even though they're actually pretty simple. Like, and you'll get you'll get the instructions step by step. So, but just wanted you to get to see all parts of it. So the next thing we're going to do for our root staining process is we are going to pour off this waste.
Do you know that those were the roots of the trees, or was it could have been some other grass?
These are the roots of the grass. Right. Yes, totally.
Right.
Yeah. Should have brought like an oven mitt, but this will be fine. Really? That could be better, actually. Oh, thanks. Are you going swimming?
Yeah.
Yeah, me too. It's really perfect.
Would it be evidence that you also would have evidence on the...
Yeah, pretty much. You usually won't have spores. I mean, the reason you would want to do root staining, obviously any experimental situation, like if you're, you know, like, because for this program, you are comparing, right? You're doing is it a commercial inoculum and then this one? And then no inoculum. So like you would want to, for any type of like study, you would want to actually check the roots because you can't always assume, especially with a commercial inoculum, that it worked, right? You could put the spores in and they're dead or something and they don't like connect with the roots. So you would want to do this root staining for that, a study kind of purposes. Otherwise, yeah, spore extraction is totally good just to make sure there's spores. There's like propagules in the soil.
How effective would warm water be compared to using the...
It's not, unfortunately not, well, oh, crap. It's not enough, unfortunately. And people have tested and tried all kinds of different modifications, but we're already doing the modified, like the chillest version of this. And especially, and it's different, like if you have agricultural roots or like, what are those called? Yeah, if you have woody roots, you really need to do these processes because it's just hard to like strip them enough that the dye will take and you can see what's going on. If you have like the grass roots we're using, you could, yeah, they probably need shorter times in each solution and stuff. Yeah.
What about like my transplant? I'll literally get the hose and spray the roots so there's like nothing on it. I don't think that could do something to
You
come
still,
close to it.
yeah, that would help. You can definitely try it. It's just still, you're still gonna have stuff that'll make it hard for like the dye to stain and stuff like that. But yeah, you can definitely try it. It just gets harder to see. Like you'll see when we look under the microscope, unless until you get experience with it, anything that's going to get in the way from you being able to see what's going on will just be a little tough. Okay, so my next step here, okay, so I poured that off. I wanted to, dang, oh yeah, yes. Tweezers, perfect. I was like, okay, so my next thing, I'm actually just gonna rinse them. So you pretty much rinse them in between each step. We also have gloves, right?
The gloves are.
Oh yeah. Cool. Cool. There we go. Perfect. It's just that these are like, this is really basic. And then the acids we're using are acidic, so I just don't want to touch them because they'll hurt my skin. It's not that they're like toxic, it's just
Does the tweezer have to be in plastic?
No,
Okay.
it would actually be better if it was. Oh, it's so hard. So I'm just gonna try it. Yeah, I'm gonna put my hands in there. Wow, this is like a fair game. You know what I mean? Yeah, that's a good idea. There we go. Okay, there we go. Solutions. Okay, and then the water. So just trying to rinse them. You're always trying to get like the last solution. Like, look how much more is coming off in there. So I'll make this, where's my wastewater? Yeah, yeah, you should be able to reuse all these. Yeah, multiple times. Give them another little rinse because they look, yeah, it's kind of wild how much is. Yeah. Okay. Okay, and then our next, oops, our next one is gonna be, thank you. Okay, cool. Next, we are gonna let it sit for 10 minutes in hydrogen peroxide, which you can just get from the grocery or the drugstore. You might already have some 3% hydrogen peroxide. You don't always need to do this step for roots like this. You want to do it for sure for like woody roots, but so we'll just do it just in case. All right, and then get my timer going, and then we'll go over to the centrifuge. 10 minutes. Okay. So there's our hydrogen peroxide, and we can take a little walk over to the. Love that. So this is what we wanted to happen, right? So we just separated all the organic matter out of here, and now we've just got our sample in the bottom. So that was the whole purpose of this step. So I'll actually hand these out. So all we're gonna do is you'll get the tape off and save it, and then you just want to pour off the water with the organic matter. So there is a little bit of a knack to it. The bottom should stay in, but I'll just show you. You just kind of, you don't want to pour out the sample. See what I'm saying? Cool. So now, while we're getting those other ones ready, we're gonna add basically sugar water. And so the recipe for this is in your handout, but it's literally just sugar and a little dash of soap. And the method here that we're doing is creating a density gradient. So because sugar is, it'll make, like, sugar water is like dense. I mean, you can feel this. It's just a jar of sugar water. And so we're going to use this sugar water to basically separate the spores in there from the soil. It's like a really simple method that works really well. And then there are a bunch of other methods that I'll talk about once we get these. So basically now we're going to fill.
The sugar water is 4%.
Yeah, it's 4% sugar water. But the measurements, it's like, forget offhand, but it's like.
Yeah.
Never use paper again.
Oh, yeah.
Is it a funnel, or do we need to make a paper funnel?
I know, yeah.
We have
I wonder.
Oh, might have some sweet fingers.
Let's see. Yeah, okay.
Yeah.
Let's see. Oh gosh. Well,
Like I said, don't mind having.
it's
some
just
sweet
sugar water,
fingers.
at least. Okay. Oh, right. We got our, do we have our tape?
It got all gone.
Okay. We can get another piece of tape on there. Cool.
Looks like we got to stop her on the way.
Oh, that's great. Oh, yeah. Oh, that's so cool.
Yeah. Great.
Nice. Okay.
Are we centrifuging again?
Yeah. First, we're going to let them sit on their side like the last time.
Should we maybe we'll use
Oh, yeah.
caps just to ensure.
For when they're, and so we're going to do the same thing. Well, if you already got it, just check it out. So we're going to shake it up like last time.
See what happens.
Yeah.
And then we'll use that in a moment. Once it's, cool.
Shake it up. And you want to like really
Make sure it's all broken.
make sure it's all mixed up. Oops. Okay, and then we're gonna lay them on their side. Sticky fingers. Oh my gosh.
Spray bottle.
Yeah, yeah, yeah. Does anyone have? Oh, thank you. Can someone set a timer, please, for 9 minutes?
Do you want to use water?
Yes. Thank you. There we go. Great. Okay, cool. Okay, cool. So we got a timer. So once they're shaken up, they're just gonna sit on their side like last time. And then we will centrifuge them. Cool.
Why sugar water?
Yeah, so it'll become more clear once we do it, but basically it creates a density gradient. And spores, the spores we're trying to extract here are of a density that when you basically pour the sample into a glass kind of like funnel, it'll separate, it'll pull all the soil away from the spores and leave the spores on the glass. So it's just kind of a simple way to separate. Another thing you can do to do spore extractions, this is more like in a way precise. So yeah, but you can also do sieving. You just have to get a bunch of specific sized sieves. And that's because spores are of known sizes for these mycorrhizae. So you can also like, basically what people will do is you take like your sample, you can even like blend it, like put it in a blender with water, basically like get it all kind of broken up and then pass it through these sieves and you have to do like a lot. It is like a little more work. You have to do a lot of like washing and sieving, but you end up also separating out specific sizes of spores that you can then store.
How much do they vary in like micron size for spore size?
I have the exact sieves you need in the handout. I think I can't remember the numbers exactly, but they're between like, what is that, like about 80 and like 300 micron. Yeah, you probably have. Cool. Yeah.
Could use a bunch of ants like that. Yeah.
Yes, please take all the spores. Yeah, the reason they're getting spores though is they do like to eat them. So
Really?
yeah.
It's like dogs with troubles.
Yeah. Sometimes they end up carrying them, sometimes they eat them. Yeah, I heard there's this, actually this is good for you to know. We talked about, yeah. In the last workshop we talked about like methods, especially for like on-farm. Oh, that's that other one. Okay. For on-farm production of mycorrhizal inoculum, and like other methods are through what's called it's called INVAM. It's like International Vesicular Arbuscular Mycorrhizal, yeah, Society or whatever. They used to be called, anyways, Vesicular Arbuscular Mycorrhizal Fungi. It's very long, but they have all these methods on. They have a bunch of methods on there as well. Basically, what I've done, like I've looked at like all kinds of different methods from different places and applications and tried to distill what I think are kind of like the simplest. Sometimes it could be overwhelming to just go and see all that, but just so you know, that is a good resource. And they also have a culture collection. So they do like really specific, like they'll extract spores, idea to like species, and then have like pure cultures essentially, because they're maintaining a culture collection. But anyways, I was talking to the person who runs the mycorrhizal culture collection for North America, and he told me that, you know, they go back and check their stocks, which are basically like little bags of sand with spores in them, just pure cultures of like by a specific species. And sometimes the spores would disappear in the culture collection, which is obviously bad, right? Because their cultures are going away. And they realized they kind of studied it and found out that it was, I can't remember if it was ants or another type of, or like some type of mite or something, but they were going in and only eating dark spores. So they would leave behind lighter spores, and they basically figured out that the mycorrhizae intentionally produce spores that are like sacrificial, like that are more noticeable in the sand or soil, so that those will get eaten and the ones that blend in better won't. That was super cool. Hey. How far are we from this? Okay. So I think what I'd say then is let's head back and do the next step over there, and then we'll get these centrifuging. Yeah. Okay, our next step. Cool. Thanks for walking with us back and forth. We're getting a little dynamic. Yeah, oh good. I'm glad. Yeah, I guess it's better than just sitting, listening for like 3 hours nonstop. Yeah. Awesome.
The white walls and always windows up high.
Yeah.
So you can't see anything. You know, prison.
Yeah. That's true. I know, it is weird. I did my undergrad at a university that was designed, I forget his name, but by some famous architect that designs prisons. And then that's a really common thing. They intentionally design them to be like, okay. So I just want to point out, I don't know if you can see, but it's good that we added this step, because you can see that we did even more clearing. There's a bunch of like bits of organic matter and like color pigment that's come out in the hydrogen peroxide. So I'm very glad we did that. So now I'm gonna basically do the same thing, pour off the wastewater. And also, just to be super safety conscious, please do not heat. Like you'll notice I didn't do this on the heat. If you heat hydrogen peroxide, it produces very bad fumes. Do not do that. I mean, outside it would be okay probably, but please don't like go home and do this in your house and heat it. You don't need to heat it for this step anyways, but I just want to be really clear on the safety info on that. Okay, and now I'm gonna do my little rinse like before. Yeah, so these processes are very similar to any type of like dyeing. You'll probably know. Does anyone do dyeing here? Anyone else? No. Mushroom dyeing. Have you guys done a workshop on that before?
We did during the pandemic online.
Oh, cool.
Artists from Oaxaca.
Oh my gosh, that's so cool.
Yeah.
I know. We just want to try to clone a mushroom. Carter's trying to clone that one, that St. Venus. Oh, yeah. I have it in my fridge. Her name is Haina. She's if you message me on the Instagram, I'll send you her profile. She now lives in Guadalajara. She's teaching students. Oh yeah.
Okay. So our next solution, cool. Would someone be down to like just go pop them in the centrifuge, or should we all go over? 20 minutes.
Take the lids off of 2 of them.
Yeah, and you'll have to tape 2 of them. So our next step is adding vinegar. The actual method calls for hydrochloric acid, which is like a little intense and harder to access, so you can just use vinegar instead. Vinegar that's about 5% acidity. And this is basically just, so what we've done so far was all to clear the roots, and now we're preparing the roots to be able to accept the stain. The reason, I should have said this too, I'm like swirling it each time because I really want the roots to be kind of completely penetrated by these solutions. Okay, so I added my vinegar, and I'm gonna do this for 10 minutes. And this is at room temperature as well. Cool, so set a timer here. Cool. And then our next step, sorry? 10 minutes. So our next step after this is we're gonna actually pour this off, rinse it, and then apply our stain. And I wanna talk a bit about the stain while we're going. Yeah, so we do need heat for that one. Okay, so the most common stain used for mycorrhizal work is trypan blue, which is like a known carcinogen. I've worked with it a lot. If you do like a PhD and work on this stuff, they will give you trypan blue to do this staining. So toxic, so bad, and you don't need to use it. So there's a bunch of papers that have explored alternative inks and stains. And yeah, the one we're planning to use today is a natural black ink. And you just, yeah, the paper actually that I sourced this info from suggested mixing the natural black ink with vinegar. Well, actually, yeah, with vinegar, but then I'll show you. I think we have that somewhere. April did that, and it just like will not mix with vinegar. So I don't know what happened there. But it does mix well with water. So we're just going to do that instead. And we're already preparing the roots with vinegar anyways to help them. They probably added it to help the stain stick. But we're already doing that step, so it's all good. And then methylene blue should work as well. Like there's a bunch of other dyes. You can use red ink. There's a bunch of other inks that will work just as well, more or less. I mean, you'll notice all the pictures you'll see are with the trypan blue. It really does give the best quality images. But these other ones will work as well. Again, we just have to kind of train our eyes to figure out what we're looking at. So I think, which I might actually talk through, because we've got 6 minutes here and probably like 18 minutes over there. Cool, thank you. Okay, cool. Just gonna double check. Yeah, okay, perfect. So now let's talk about what we're gonna be looking for, because we're getting closer to the steps of getting to all look at this stuff under a microscope. So I'm gonna bring this back. Okay. Okay, so let's start with ectomycorrhizae, which we might not see. We might not see ectomycorrhizae, but it's also possible that we will. Sometimes you can actually see it on the outside, but what we're going to be looking at is the internal structures of the root. So this is a microscopic image of what is called the mantle for ectomycorrhizal fungi, and that's just what it looks like. It's like a fuzzy, it almost looks like roots, but it's actually fine mycelium that is on the outside of the root cell or the root itself. Then we have, yeah, so that's like the fungal sheath. This is the essentially hyphae, that like the thread of fungal mycelium that's growing inside the root around the cells. It looks like someone almost took a marker and like outlined all the cells, but in like sometimes a random path, like however it wanted to weave its way through the plant cells. That's called the Hartig net, I think, because some guy named something Hartig, noticed that. And so that's like an easy way to distinguish ectomycorrhizae from the AMF. The AMF structures we're looking for are all shown here. So these little, like, somewhat tree-like looking branching that are right inside the cell, the plant cell, those are called arbuscules. That's where the trading happens. Sometimes in a lower quality image, it can just look like a little blurry, almost like cluster. Like there's a bunch, yeah. And that's different from when you're seeing vesicles or spores. So vesicles are these cool little structures that arbuscular mycorrhizal fungi form. They can be a different shape than spores, which tend to be like very spherical. I mean, there's different types of spores, but generally if you see like a glowing like orb, that's probably a spore. They can produce them inside. Typically they will produce them outside. Some of the processes we've done can like kind of strip away or like separate them a bit. But usually you would have I'm trying to see if there's a picture of this. Usually you would have like a little thread of hyphae and then a nice big spore outside. And then the hyphae can look similar to how you see the Hartig net for the ectomycorrhizae, but they're not as neatly outlining the plant cells, right? So you can have hyphae, remember, because AMF, I should have said this, they're also called endomycorrhizal fungi. So if you go to buy a commercial inoculum, you probably won't see it say like arbuscular mycorrhizae. It'll probably say endo or ecto or endo-ecto mix. Exactly. And that's why, because AMF go on the inside. So you can see their hyphae. Sometimes they're outlining, like they're growing between the cells, but then they'll also just go right through the cell, the plant cell. Another cool thing, as I mentioned, I've done a lot of work on contaminated sites. And something really interesting is if you're looking at mycorrhizae from contaminated sites versus ones from like a healthier system, you're going to see a lot more vesicles inside when you're looking at the roots. Can anyone guess why that might be? More vesicles in the contaminated, in the mycorrhizae growing in a contaminated site, which you're onto something with that. Yeah. Yeah, vesicles are like these little storage compartments that they make, like little closets, and they'll just shove stuff in there. And it's really cool. So you'll see, yeah, just a lot more vesicles, and people have looked into it and they'll find, so like metals, right? Again, metals don't break down. Sometimes the arbuscular mycorrhizal fungus will take them, like they can increase uptake in their plant, but remember they're living like partially inside of the plant roots. And so they'll take them in, bind them with phosphate. So if any chemistry people will get that interaction, but for lead and a few other types of more toxic metals, they can be like essentially bound up, trapped in place when like a phosphorus is added. So they'll add some phosphorus, chuck the lead in this little vesicle storage compartment, and then keep themselves safe, keep their plant partner safe from that lead, from that toxic lead.
Does it ever break down the lead, or is it more of just storing in it and just having somewhere to put it?
Well, because lead can't break down because it's elemental, so it can be transformed or moved around. So this is a really clever way for the fungus to deal with it. Super cool.
How that whole system understands what is harmful and what is not.
They are brilliant. I don't know, but I know they know more than us, I think, because they do just wild things. And I mean, I've been told, like, I've studied fungi, but I really love mycorrhizal fungi. I've studied them for a long time, and I've had other like people be like, why do you study them? They're too complex. Nobody understands them. We just got to, you know.
That's why you study.
I was like, that's what's so interesting. It's so interesting. But like another funny example is, I'm just going to set this aside for a sec. For example, we thought that they were asexual, like meaning that they basically produce, they don't like mate essentially with another, or mycorrhizae. They just like do their own thing and produce the spores that way. But then somebody basically discovered that they have the machinery, so to speak. How else could I say that? They have all the bits to like sexually reproduce. And so what that says to me is that they just don't like having sex in front of human researchers. You know what I mean? Like they so they're just very mysterious. They've got all kinds of mysteries going on. I love it. Sequencing is very cool. We can learn a lot from it, but like they kind of resisted. We only recently were able to even sequence a genome for arbuscular mycorrhizal fungi. Before that, they were like difficult. Like we couldn't even figure out how to like learn about them. So they're very mysterious and very cool. And private. Well, but they're out there in these networks connected to literally like all plants on Earth, pretty much. But then, yeah, with human, I don't know. They're like, don't get my genetics captured. We don't know what you're going to do with that. It seems to have not worked out well for some other organisms. So I don't know. So basically, weird maybe answer, but I don't know how they know stuff. But they seem to know a lot, and they do wild things. Like they also get predated upon by, like I said, like different insects and stuff. I saw some studies that, or I don't know if you know, but in agriculture, we've used copper and arsenic as insecticides and pesticides and stuff because they're toxic to certain pests. They're also toxic to these fungi, but some of them have been observed essentially collecting copper and arsenic which is toxic to them too, from the soil environment. So scavenging, grabbing onto the copper and lining it on the outside of their mycelium to protect them from predation. So they're really smart. Yeah, no. Yeah. So I'm just pouring off this was the vinegar. I'm gonna rinse it, and then we're gonna add our dye on the heat for a bit. Okay. So, all right. We've rinsed with vinegar. We've poured off, and now we are going to stain. Make sure that's fully covered. Yeah. Okay. And I'm gonna put this on the low heat. Okay, cool. And then I'm gonna set my timer.
That also be 90 degrees or?
Yeah, exactly. Okay, cool. And then do we have a time check on the? Okay, cool. Oh, perfect. Okay, so our next steps. 5 minutes. You might need to do longer if it's like really woody roots. Okay, so we're staining in ink for 5 minutes, and then we're gonna destain, and then we are gonna go over there and finish our workshop looking at stuff under a microscope. So yeah, okay. So we've got about a 5-minute period. Do people have any questions at this point while we're waiting for our stain to take hold? Yeah. Yeah, that's a good question. Yeah. Yeah, so the question was about the vesicles, right? That, or just like in general, is there a capacity for storing and dealing with toxic stuff that these mycorrhizae have? And the answer is yes, but it really depends. So some species are more sensitive than others. So if you like looked at, again, like a healthy, diverse ecosystem, you would have probably different species and more diversity than if you looked at a contaminated site. It would kind of filter for only the ones that can tolerate that level of toxicity. So yeah, some are more sensitive than others. And then within each, it's really hard to even know what an individual is with mycorrhizae, but within like a mycorrhizae, it will have a certain capacity. So if things get like more toxic or whatever, at some point it's going to start to be impacted. And so will the plant. I mean, the same is true for plants. There's only so much they can tolerate, but they can adapt also over time. So, but that tends to work best when they're slowly, like they have time to adapt. If that makes sense. So if you just like dumped a whole bunch of, like if it was all fine and then you dumped a bunch of toxic stuff on them, they might not have time to adapt and they might die. And then your question made me think of something I wanted to share too. Like these relationships are extremely dynamic. Not everything is known about how dynamic they are in soils underground, but it's thought that like a single plant, so like a tree right here, could have a bunch of different arbuscular and maybe ecto and maybe other types of relationships underground, and those all might be forming, dying, reforming, then another spore germinates, it reforms again, like on a daily basis. It's like a very dynamic system. And the capacity for each plant to form these relationships is essentially like as much as there is space in the roots. Yeah.
The mycorrhizal fungi, do they have relationships with other fungi? Do they compete? Do they cooperate?
Not too much. That's a great question. I don't know if that's really understood very well. But yeah, they seem to, I mean, they're basically in there figuring out how to relate they're all figuring out how to relate to one another. So generally, like a mycorrhizal fungus is not going to be competing with like a decomposer because they're getting food from different places. So they can share the same, they're occupying different niches, right? One is like in the roots, one is growing on the organic matter in the soil. They can be weaving their mycelium together and getting food in different ways. Yeah. Oh, that's a good question. Yeah, they're all thought to be. I mean, AMF are generally generalists. So yeah, they're not specialized in that way. The other types of mycorrhizae are, but there are some. But it's also interesting because even like the plant families that are thought to not form mycorrhizal relationships sometimes do. Like, and if it's just that not that many people are out there like looking, right, like doing what we're doing today, and so sometimes like I've seen brassicas that had, maybe they're not going to have the full root is like taken over by the mycorrhizae, but they might have a little bit. Yeah, but yeah, AMF are generalists. Then there's like orchid mycorrhizae that are really specific only to orchids, and then the ectos with like, yeah, coniferous trees. But like I said, even coniferous trees can be arbuscular mycorrhizal earlier in their lives. So yeah.
Consider use both.
Yeah, totally. All the time. Yeah. Mm-hmm. Yeah. Mm-hmm. Would be AMF. Yeah, grassland would be AMF dominated, and then the forest would be ecto dominated. But at the same time, it depends on the age and stage of the forest. Earlier on, like those trees probably got their start with arbuscular mycorrhizae, and then those relationships fall away for the ectos.
Is there a survival reason for that? Like why they do better with the AMF first, and why later on they'd go to the ecto?
Yeah, let's think through it together. I mean, folks that work with plants or like have nurseries or anything, I mean, do plants, I'm trying to think if like a pine tree, say, would have different nutritional needs or other needs later in its life versus when it's a sapling, like when it's just growing. Could be something like that. Any other ideas? Yeah. Oh, that's a really good question. So what I've learned is that the plant essentially dictates the relationship by putting out sugars into the soil to attract certain. So it's the plant must want these different relationships at different times. I'm not sure why. Yeah.
Like fruiting and flowering versus leaves.
Yeah.
Versus roots.
Yeah.
Your NPK and each one.
Yeah, exactly.
So maybe when it gets mature and it's trying to make pine nuts or trying to make fruits.
Yeah.
Need different things extracted from the soil.
Yeah. Yeah, totally. It definitely could be something like that. Yeah.
Is there a difference maybe like in the depth to which a plant can communicate these needs to the fungi?
Hmm.
There's a difference, like if it was raised in a sterile environment without the fungi and then it was introduced later versus if it grew up with the fungi. Like, does it like learn how to communicate earlier? Like,
that's such an interesting question. Wow. I don't know. I don't know, but I love it. Yeah, I love that question. The question was about like how do they learn, like kind of like, yeah, do they learn, like how does the plant know to put out these sugars and stuff? That's like it makes me think of like coevolution. It's kind of trippy to think about, especially the species that like only, like they each give up like really important like things essential for their lives so that only one specific, like they they need each other. Like what is that? Is it like cashews or something that only are germinated by like this one type of wasp that's whole life it's figs, that's what it is. Yes. So like it has to be some type of coevolutionary thing. So much more communication going on in nature than we can currently seem to pick up on. Oh, there was something else I wanted to share, but I'm forgetting it right now. But yeah, I love this discussions.
I'm just curious about the orchid thing.
Oh, yeah.
The mycorrhizae only used for the germination of the seed.
Yes.
I was wondering because they're air plants, so I was wondering how
Yeah.
mycorrhizae would
There's
grow.
some that grow in the soil too, or native ones in the soil.
That's true. Yeah, I
Yeah.
only deal with I grow orchids, but only exotic species.
Yeah.
I'm just curious. I've always wanted to try growing them from seed.
Yeah, totally. Yeah, it's super cool. Okay, let's sort out our dye situation. Okay, so we're basically going to rinse them. Okay, let's make sure this isn't too hot. Cool. Okay. And this dye isn't toxic, so that's nice. Get another waste container. Oh yeah, that's what I was gonna say. There have been some studies about how mycorrhizal networks seem to facilitate succession. And by that, I mean like this maybe transition from a grassland into like a complex forest, like old forest. And basically the reason that's been observed and like hypothesized is because there's been studies that have tracked either with like isotopes, like carbon isotopes, basically tracked the transfer of carbon through the mycorrhizal networks between different plant species, and observed a pattern of like at a certain point, Dennis was just talking about alder trees. They're like an early successional tree. And there's been observed that at a certain point, the mycorrhizal network, for some reason, stops sharing nutrients and stuff and transferring carbon from the other trees to alders. And then they start to like get create space essentially in the canopy and in the soil for longer lived trees to start to come in. So like, why is the mycorrhiza, I don't know, but it's really interesting how they seem to facilitate like an ecosystem level of change, I guess. Interesting. I don't know. Yeah. That's a good point. You're like, better sugar over here. So and if I didn't say that, it's literally sugar, is carbon. That's really what the mycorrhizae is getting. Again, it only lives based on carbon shared by their plant partner that the plant produces through photosynthesis and then sends below ground to these. That's their part of the trade. Yeah. Better bet. Yeah. That's a good point. Yeah. Like it's a more stable relationship. Yeah. 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 Cool. So I'm just no situationships. Not preferred anyways. Yeah. Yeah.
I was thinking exactly that.
Well, do you mean like in evolutionary history of Earth?
We run into we deal with a lot of like construction soils.
Okay. Yeah. Mm-hmm. Yeah. Okay. Yeah. Yeah. That's a good question. Thanks for bringing that up. So whether you're using a commercial inoculum or inoculum you produce like this, the time to apply it is when you're planting. whether sowing seeds, you literally can like mix together the spores or the inoculum, like the soil inoculum, like in a bucket, and then plant your seeds. Or if you're transplanting or planting a tree or something, you want to like, dig your hole and then put the inoculum in the bottom of the hole and then plant your tree. And that's again because these are root associated fungi. They need to be in close enough contact with the roots to get to germinate and start to grow. And then in terms of like what came first, it's like hypothesized that mycorrhizae helped plants come onto land, and that they a long time ago developed a coevolutionary relationship where plants basically gave up. They were like, okay, we're not going to develop extensive root systems. We will develop this relationship, which will mean we invest less energy into building roots, which would take a lot of energy, and instead do a trade with mycorrhizae, who greatly expand their root zone and ability to get water and nutrients and stuff from a greater area. Okay, so do you know, is the centrifuge done? Okay, cool. So let me just say, so basically now we're gonna go start some microscopy. So, but I could use some help bringing a couple things over. You can have a chance to look. So all I'm doing is getting the cassettes ready, and then we're gonna start our spore extraction. Just need something to catch the, maybe like, that's not, so maybe I'll set it here. So basically, I just kind of, good call. Yeah, that's a really good idea. Where is the, oh yeah, nice. In the meantime, can I have people starting to take these tapes off the top? Start to get the tape off, please. Cool. Okay, nice. So, oh, nice. I like this. And so this is called a separatory funnel. You can also do this with just like if you have a glass funnel of any kinds, really. But let's see. I don't know if kind of get the cooler wet. How do I do this?
Need that duct tape again.
Duct tape, so handy. Okay, nice, cool. So this bottom part is called the stopcock, and it has a hole in it. So you basically just want to, and then this is the top. So basically all we're gonna do is pour our sugar solution in. Okay. Do you want to pour that one in? Cool. Thanks. And then you want to pour the last one in. Cool, thank you. And then we're just gonna set it to drip slowly. And again, this is creating a density gradient. And what it should do is basically leave all the spores on the glass walls. This little bit of soap we added basically creates a charge for the spores that should help them attach. And then you want it to be dripping really slowly. Okay, that seems good. And while that's going, we will start our looking at these roots. So basically, we just want to, we can either put these on a slide. Let's do that. And then if we turn the computer around, then maybe everyone can like be able to see. Hopefully, okay. It's all set up. Okay, nice. Oh, that's the covers. Cool. There a little this thing. That's great. Hopefully, yeah. That seems fine. Yeah. Okay. So I'm just gonna get, yeah, some little pieces of these roots. They don't look super dyed. I hope we can still see stuff.
Do you think it's because of the vinegar mixture?
That's weird.
I was reading that certain inks do not take on vinegar, but some of them do. So I think it's just this India ink.
Yeah. So I'm just gonna look and try to find a more dyed roots. So hopefully, so the ink is supposed to only bind to the fungal structures. Oops. Let's see.
Why the ink only binds to the fungi? Is it like
It's a chemical, yeah, a chemical thing. Because like fungal structures are have a lot of like protein and stuff in there. Dang. Okay.
It's barely dripping. I don't know.
Yeah, that's good. As long as it is dripping. There we go. I'll do it a little more. Okay, let's see. I hope there's. Dang.
Oh.
Well, you know what else we could do? Someone grab the methylene blue, and then we'll try that. And just a jar would be awesome. We'll still take a look at this, but we could always soak it for a few minutes in methylene blue and see what we got.
Do you see any dyeing at all? Maybe
It's hard to say. Like it just didn't really take, which I've never seen before. It's so weird.
It's this ink that I got. Did she say it's the ink? Oh. Maybe it's the ink. Maybe it wasn't.
But if it only goes with the spores and these are not visible to the naked
possible
eye,
too. Yeah.
How would
or
you see
it just doesn't.
There's
There's so many different
just
types of
because there's that
natural
many
ones. I was trying to find the most.
Oh yeah, because we're
Yeah.
separating the spores. Okay.
So
Okay.
I don't remember what this is. From a plant.
I'm just playing around at this point. Let's see what we can do here. Yeah. Science is basically fuck around and find out. Yeah. But okay. But then with a method. Like not randomly, but like where you're like, oh, I wonder if
process based.
Well, let's check this out. We'll see. I don't know if we'll be able to see much on here. But dang.
Bring a control.
Let's see.
I put a lot of that India inks.
Is it?
Like ones that are used for collective
Yeah.
stuff are all like insect based.
I think this one is. That's what it is.
Is it that the?
No.
Definitely not.
And they're saying that they collect it because the insect leaves this on a tree instead of like taking it directly from the insect.
So it's not there.
It's not working.
Should it be? Oh, so yeah, I was like, why is it so dark? That's
Yeah, and I'm very confused by that.
yeah.
Why are you so
Because
dark?
it's like, do we have the other microscopes too
Me too.
as a backup? Okay,
Me too.
with the
I wonder.
yeah.
That's the Instagram microscope.
So if we, this is the one I always put it on, so I don't know what's going
Let's
on, but we
try
can
it.
let's try it. That's interesting.
Yeah, the left one has the corrected lens.
When you removed it, it goes bright.
Yeah, but it seems like it did. Yeah, maybe I hadn't. Maybe I switched it around and didn't.
But
I
it's
never put it
still
back.
like so difficult to.
Oh, it's on the oil immersion lens. That's why.
Oh, oh
So
my
we
gosh.
could just move the over to like the 40 or the
Okay, got it.
whatever one. That's why. That's
Okay.
what's up. We just need to switch the lens out.
But then
Hopefully,
it's still so kind of funny to use it this way, backwards, but Oh, okay. Oh my gosh, there we go.
Wonderful. I can work on this too, so that
Yeah, let's do that. Okay.
we have something.
And still.
Where are we there?
Okay, here we go. Here we go. Let's see if we can see any. Oh, okay. Okay, it stained a little bit. So let's try to see what we can see here. And is there a... Okay, cool. So, oops. Oh. I lost it. Okay. Oh my God, that is cool. Do you see that? What does that look like to you? That looks like a vesicle to me. Super clear too. Okay, so it kind of worked. Yeah. And so what you can actually see, it worked. Yay! So basically what I would say is happening is that's possibly a hyphal strand with a vesicle on it. So this guy is doing some storage. Oops. Oh, this turned out better than it seemed. It's still not as pronounced, but I'm just gonna take a look here. It's definitely yeah. I think too, it's hard to see because of the it's hard to see because of the stain isn't strong, but I think you can see some hyphae and some little arbuscules. They're kind of like, do we have the pictures? Yeah. Yeah, do you see what I'm saying? You see how, and you see like it could be a vesicle, but you have these little areas of kind of like a branching. Oh yeah. Oh, that was even more clear. Yeah, sorry. Let me get out of the way.
Here too, a little bit. We can kind of maybe turn this.
Yeah.
That way. Maybe that way.
Oops.
I was trying to get it connected, but
Oops.
it creates its own Wi-Fi. Yeah. Did it come up?
Yeah, so that...
I have a question. You're looking at a root, right? Or are you looking at a spore?
It's folder. Did you see a light? Oh, right here, this light. That big old light.
The Amscope has the XY.
Do you like spot down?
Sorry?
It has the fine.
Oh, down here?
Yeah, yeah.
Oh my gosh, that's way easier. Thank you. So I'm just trying to. Oops. Okay, cool. Oh yeah. So yeah, you can see it's faint, but you can see the fungal hyphae kind of almost randomly just doing what it wants through here. And then I would say that these are probably arbuscules. Yeah, and you've got a few of them going on. So, oh yeah. Okay, cool.
You just see it.
So those like ghostly outlines are hyphae.
Yeah.
So would it mostly just be white if nothing took?
Yeah, you would just see the plant, some of the plant structures like really, really faint. But yeah, anything that is like, yeah, it is ghostly. It's a good description. Is like the stain of the fungal. Such a bummer that like toxic stuff is so much better. But you can definitely still see there. It looks like there's a lot of infection. And if you're like doing this for a study or something, you can use like a grid line intersect. You make a grid on your and you can actually quantify. You can be like, oh, this root is like 50% colonized or infected, or
Dark line.
yeah.
Would that be mycorrhizal or?
That's what I'm wondering too. It's just hard to see, but it
doesn't have any little things
coming off of it. I know, but those can get removed in the staining process sometime. Okay, I want to go.
In other words, it could be both AMF and ecto.
Definitely. And I think for the species we were looking at, they do form both.
Okay.
Yeah, that's a really
good.
good.
The grass that we use, the rye.
Yeah.
Okay.
Oh yeah, but we originally collected from oak and
pine.
yeah.
And,
but yeah,
and
no, these should be AMF actually, which I think is what. Oh, there's a pretty good arbuscule. How do I get over there? Oops.
On the lower left.
Yeah. I wonder if I can increase the. Yeah.
You're looking at, oh, this is this. I did this to students all the time. Obviously, you can see this. Well, there's no.
You're like, no.
Coming around, don't you? You are
What are
such a cutie.
Oh, nice.
Look at you, love. Oh, little one. You didn't want to be on my sweaty
Oh,
skin
cool.
before. Now you're okay with it.
Ay, ay, ay. Sorry, it's weird to do this. Oh,
I
cool.
want you to not be on the table because we're gonna.
This is kind of cool. You actually can see little like extra radical hyphae or mycelium there.
Okay.
I know it's kind of blurry. Let's see if I can. Oh, heck yeah. Okay. So I realize this is still really faint, but if you compare kind of like what we're seeing here with some of the pictures, you can definitely see the mycorrhizal hyphae growing through the root. You can see there's like little arbuscules. You're like, no.
Now I'm like, wow.
No, it's
this
really
is
hard.
So good.
Yeah.
No, I am just in shock right now to see them.
Yeah, I can't tell. Let me see if I can
Quite
focus a little
unbelievable.
more. Oh, cool. Oh, heck yeah.
Look at that. That's
Yeah.
unreal.
Yeah. Okay, you can, yeah, you can actually see it pretty well now. Okay, cool.
So can you point what you're seeing?
Yeah.
So we can see.
Yeah, so basically where you're seeing like lines running like the length of the root, and then they're also branching out. That's the hyphae. So that's like the little highway for the arbuscular mycorrhizal fungus. And then where you're seeing often where they're branching to, it'll be like a little blur. I don't know how else to say it. How would you describe? Like those are the arbuscules. So it's like the trading center. So it kind of looks a little like if you see that picture. So basically these roots have, I think we saw one vesicle early on, but I haven't seen, these ones have like almost no vesicles, which makes sense. They don't maybe need it. We might be seeing, I can't tell if that's a spore. Okay, cool. Thank you. Yeah. Actually worked a little better. We could still check the one that we just added the blue to see if that's even better.
Maybe if you, I hate to say this, but maybe we can kind of rotate in to get a
Yes, great idea.
Yeah.
Oh, okay.
So just And are there low-tech methods to find out what species of mycorrhizal these are?
Our eyes.
Yeah, the
Yes, yeah. Just and so I printed out a so what you would want to do is like print out a picture of what different spore types look like for different species, and then literally just compare and be like, oh, so like something like this that you can find, and you're like, okay, yeah.
Wow.
Is it pretty easy to tell them, like classify by species, or?
I think so. I remember when I started, I was like, this seems so hard. But it's like with mushroom, it's like with anything. Yeah. Over time, you're like, oh.
And these are so
like Gigaspora is gold and always perfectly round.
Kansas, or the
I just printed this from offline. Yeah, you can. Yeah, there's resources online if you just kind of look like spore or mycorrhizal spore identification.
But it will be contingent upon like your locale. How many do we know? How
Yeah.
many species do we know? Okay.
Oh, good question.
Right, and do we know a lot
few-
in Texas?
Well, that's what kind of why I was curious, because I was like, I don't know. I'm curious what kind of mycorrhizae are here.
There is.
Yeah.
Yeah.
Yes, and so we can look at those next.
Do they like by genus? Do they kind of like look similar?
Yes.
Sometimes? Okay.
And it becomes really clear. You can see they have kind of like different shapes, different colors, different sizes. I want to give everyone a chance to rotate through before we. Pardon? Yeah.
They're all like almost look like they're squished.
That's because they got squished. Yeah.
Okay.
And they burst.
Okay.
Okay. So I'm gonna prepare. Oh gosh, our spore sample now to just show you how to do that. So what you would want to do is close the stopper. I want to get something under there just in case. And then you're just gonna use, it's hard to do, you need some kind of spray bottle to really get, like wash down the sides. Because remember, we should have caught all the spores, basically separated them from the sugar water on the side. And so now we're trying to collect them. Like we're getting them off the sides. We've got a lot of organic matter, which is fine. And then we're just gonna get them into a little dish so we can look at them under the microscope. So, oh, but something else people is good to do is to actually like shake it and make sure you got them all off.
And this is just DI water.
Yes. Oh, it's just this one. Sorry.
Are you gonna take it from like that?
Yeah. There we go. Okay. Cool. Oh, it's now it's stuck under there. Yeah. Okay.
I put this on at the bottom, so I think
There we go.
There we go.
Okay, cool.
Wonderful.
Okay, cool.
I mean, if you don't get your nose on it.
Okay. Just trying to make sure I really got all the spores off the side. Can somebody get a few more of those petri dishes, please? Cool, thank you. Cool, let's see what we got. Can somebody else kind of keep that going? Trying to get them all out. And then I'm gonna take a look, see what we got. Okay.
For like a sample from a single plant, how many species would you like expect it to associate with?
Oh, so yeah, I've done this a lot on contaminated sites where there's lower diversity, and I think I had an average of like 7 different species per plant. So a healthy system could have, you know, more than 7. Yeah.
And do you know, like, if you know, because you really were emphasizing the bioregional aspect of
Yeah.
Now it's just
like within the data sets that are out there, are they layering in the geolocation to see if there's like how the diversity changes throughout?
Yeah.
Is that
Yeah,
very
that's
there?
kind of what Spun is doing, I think.
Yeah.
So, but I don't know. Has anyone seen if Texas has been?
I know
Has like
someone's
Central?
told me, like the person that owns the Wild Root Organic, which was the mycorrhizal,
Oh, yeah,
commercial
yeah.
mycorrhizal inoculum that we use for the phase 1 of the trees that we planted here. He said that in the 90s there was a survey done for Texas and actually all through
Oh my
the
gosh, cool.
Southwest.
For
But
mycorrhizae?
I don't know where that data is. I'm sure
Cool.
it's like locked up somewhere in some university and funding was lost.
Yeah.
The
Oh my
usual
gosh.
case.
Yeah.
It's like it was in the 90s, so we don't know.
So this is, I'm gonna have to get one with less